Hey, everybody. Look, it's amplified sound. My name's Todd Runstead. I'm the content director for naturalproductsinsider.com. Go there for your daily informational needs. Welcome to SupplySide East. We've got a really good program this morning right here. We've got two experts in the microbiome. Yes, we called it the Maxobiome because we're clever that way. And so a couple things. First of all, if we can flip the slide, you can see, here's some show announcements, but then there's also this QR code that you'll want to scan. So I guess there. This is the moment to break out your phones, scan the QR code. And what this will do is we have this a live real-time Q&A thing. So if you want to ask questions of our speakers, you can type them in to the Vvox platform that is right there. And so if you do that, you can see on your phone, this is way too small, but you can get there if you scan the QR code. And then there's the little Monopoly house on the upper left, and that's this program. And the middle one is Q&A, and then the one on the right is a survey. We would like to get your comments on what you think of the show in the next hour. And once you do that, then you can avail yourself of the slides. And so we're tricky like that. So anyway, yeah, that is, I guess we're jumping right into the first question here. What health target is best addressed with probiotics? And and it looks like digestive health is in the lead by a healthy margin. Immunity coming in second. The gut-brain axis is third, but asking 5 years and the gut-brain axis might be the winner. All right. That question's over. Hey, can we go back to the, before we go to question number 2 of 3, I want the housekeeping announcements. There's our fellows. Can we go back one more? There. Little bullet point number 2 is the Wi-Fi password. Everyone wants the Wi-Fi is SupplySide East 24, and the password Ingredients Online, which is no doubt a sponsor. And the expo hall is open till 4 today, 3:30 tomorrow, because, you know, bankers' hours. There's also a food truck outside. Oh, and then there's a couple cool things. I know there's a bunch of you who are first-timers here, like more than half the people who are here, so righteous, get your education on. Accelerating newcomers in the industry is right after this thing at the SupplySide Connect Hub, which is back and to the left, just like where every restroom is in every restaurant, back and to the left. So that's the SupplySide Connect Hub, and so meet a couple of your fellow newbies and learn a little something. We also have a happy hour today on the show floor from 2:30 to 4, because why not? And and then this is also fun at the SupplySide Hub, Connect Hub. Also, at the end of the day, is the speed networking. And so if you want to meet fellow friends, meet potential leads, all these fun things, again, back and to the left. And then there's the What's Up with Sups party. Now that's a good time. And if it's anything like the last couple of years, it's a fabulous view over the Hudson to the Manhattan skyline. Lovely place. Yeah. Okay, so now let's flip ahead, shall we, to the, let's see, there's a QR code to scan again. I think before we introduce these guys, let's go to Vvox question number 2. There it is. What is the best way to succeed in the microbiome business? Question 1, clinically validated doses of specific bacterial strains. Strains matter. Oh, look, one person's voted. 2, find the next Jamie Lee Curtis. Come on. And what, 21 million? That was like the fine that they had in their P&L in order to, make those untoward claims. Yeah. And then they, Dannon made 100 million, so that was a good investment. To add a 0 to your company's valuation, add microbiome to your company name. That's a clever one. And then we were just talking about this before. Number 4, hire savvy marketers and influencers. Wait, I thought influencers are free. Is that not? We're going to find out the answer to this question. And yes, it looks like, look at all the responsible people here, clinically validated doses of specific bacterial strains. But what about prebiotics, people? What about postbiotics? Those aren't strain specific, are they? We're about to find out. We're about to find out, people. Okay. Question number 3, our third and final one, and your blood better be flowing at this point. Let's see. VVOX question number 3. What is the best way to formulate a microbiome supplement? A, it's all about the CFUs. The more the merrier. That's right. If it's not 50 billion, you're just a supplement. 2, probiotics need prebiotics. Everyone needs a lunchbox, right? Number 3, specific bacterial strains are where it's at, right? That's what we voted for last time. And number 4, complementary non-biotic ingredients. Ooh, that's a savvy, sophisticated formulation going on right there. And going once, going twice, and it's looking like specific bacterial strains are where it's at. Thank you. Thanks for playing. Thanks for being here. Now I'm going to introduce our 2 luminaries here, sitting here in the front row. Coming up first, we have let's see. Yeah, we're going to read left to right here. We have Noah Zimmerman. Now, he is the Chief Science Officer of Verb Biotics. You can read all about the visit that I took to the Verb Biotics labs down in the Research Triangle in Raleigh-Durham area. Super cool little outfit that they've got going on here. Noah is also, in a previous life, an academic of University of Wisconsin Hospital. I might have those words a little mixed up. Medical College of Wisconsin there in Milwaukee. And he's a sharp fella, and we're really happy to have him here. And then, so he's going to talk for half of this session, and then we're going to get into the business strategies side of the biotics business. Say that 5 times fast. So without any further ado, Noah, come on up here and tell us something we don't already know.
All right, thanks, Todd. So I'm Noah Zimmerman. I'm the CEO, or CSO, CEO maybe down the line, the CSO of Verb Biotics. I've been studying gut health and the gut microbiome for the last 2 decades. And so during that time, what I've realized is that the bacteria that are in your gut aren't just kind of sitting there clumped together. They actually function as an organ. And a lot of times that organ may be forgotten or really maybe just underappreciated. What I'd like to do today is just make sure we're all on the same page. So I'd like to start off by talking about the gut microbiome as an ecosystem. I'd like to talk a little bit about how it's interconnected to other organs throughout our body and has an impact throughout our entire body. And then I'd also like to talk a bit about biotics and how we can start to develop truly functional biotics through using some of the biotechnology and science that we have at our hands right now, because this is a really special time for biotechnology. So starting off with the gut microbiome as an ecosystem, we look at the microbiome as being incredibly crucial to overall health. We look at it as an organ, similar to the gut, or to the brain, to the heart, to the lungs. And you really can't live a healthy life without a strong microbiome. The vitality and the diversity is really linked to our overall human health. So a really good way to think about your biome and your microbiome is thinking about it kind of like you would think about a jungle. You know, in a jungle, you have large keystone species that grow. Typically, these are large trees, and they create an environment where you can have other things start to come in, like flowering plants, followed by pollinators, and then maybe herbivores come in, and then predators, and everything's in a nice balance and functioning pretty well. And the microbiome is very similar to that. There's a lot of diversity in your microbiome. A lot of things are interconnected, and you need to have kind of all the legs to that stool for it to be nice and healthy. Now, just like a jungle as well, if you look at a jungle in, say, South America, it may look similar but be very different than a jungle that you would see in Africa or, you know, say, Southeast Asia. And that's true of your microbiome as well. So I have a specific microbiome, and mine's different than Mike's and Todd's, definitely, and different than everyone else in here. And so we all have this biome that functions differently. The opportunity there is that we are processing foods differently, we're processing supplements differently, we're processing some of the pharmaceuticals that we take into our body differently. And I think one way that this is always illustrated to me usually is around, typically it's Thanksgiving, but family get-togethers, where it's usually an aunt or a cousin has some new diet that they're trying. And that diet works fantastic for them. They can sleep well, they have a lot of energy, they lose 50 pounds, and I think to myself, I'd like to try that. So I try that diet, and I get no benefit at all. And when we dig into this and we look at clinical trials where this has been looked at, what we find is that the reason that it worked for those people is because they have specific microbes that are able to break down and create specific metabolites that give them some functionality. And maybe I don't have that in my microbiome. Now, you can look at that as a challenge, but we really look at that as an opportunity. What this allows is for us to go in and study those people. What is it about them? What is it about their microbiome? What bacteria do they have? that can provide that benefit? Can we take those and provide that to the overall masses to shift everybody to have a healthier gut? And the answer, in my opinion, is yes. So what I'd like to do is talk about, what is the microbiome? The microbiome for the gut is home to over 1 trillion, and actually more recent estimates are closer to 100 trillion different microbes. Typically, we talk about bacteria because those are the easiest to study. We have the most tools for that. But there are a lot of other microbes as well. Things like archaea, which are a bit more ancient organisms. We have eukaryotes. There might be amoeba or other things that are swimming through your body. We also have pathogens. And then there are a lot of viruses as well. Oops, we'll go back one. And the other thing with this microbiome is that today I'm going to talk about the gut microbiome, but there are lots of different microbiomes. There are specific areas of the body that have different subsets of microbes that perform very important physiological functions. You have a nasal microbiome, an oral microbiome. Your skin has a skin microbiome. I'm going to talk about the gut microbiome because this is really the highest concentration of microbes that we see, and it's a major interaction between a lot of the substances that we take up in our body. So what is the microbiome and what does it really do for us? Well, the first is that it aids in food digestion, and it can do things like digest fibers and release nutrients. And while that sounds kind of bland, like maybe it's just giving you some sort of energy so that you can grow and go about your day, the microbiome is able to interact with everything that you're taking into your body and actually release a treasure trove of different bioactives. So things that are being released don't just stay in your gut. They circulate throughout your entire body. They can have impacts on your brain. They can have impacts on your skin. Everything from your muscles to your immune system. So this is all being released by what's happening in your body, and your microbiome kind of dictates how those microbes or how those metabolites are being made. We also know that the microbiome produces some of the essential vitamins. Things like the water-soluble B vitamins and vitamin K are exclusively made by the members of the microbiome. And then it also helps to develop and regulate the immune system. It is very important for training the overall response of your immune system. So early on, when your body is starting to figure out what is normal, what is new, what is something that I should watch out for, the microbiome is directly involved in training that immune system. And those immune cells don't stay just in the gut. So it's not Las Vegas. What happens in the gut happens all over your body. So those immune cells, once they're trained, can leave, and they can go to different parts of your body, and they're continuing to surveil the areas to see what is new, what's normal, what should I watch out for. And then finally, the microbiome is also involved in preventing pathogens from taking up residence. And it can do it through several different ways. It can directly kill off pathogens, or it can just go in there and outcompete them and steal the food that they're looking for. So that's in general what the microbiome does. And I think if you think about what it's doing with the immune system and metabolites that are being made and how they can circulate, it's pretty clear that it doesn't stay within the gut. What happens there can circulate and have interconnectivity to other areas in the body as well. And I think this is well illustrated when we look at something like disbiosis. So disbiosis is that imbalance of the microbiome that's associated with disease and with inflammation. So typically in a normal, happy gut, what you have is a single cell layer. So these are the intestinal epithelial cells that are growing nice and tight to each other, and they have little connections called tight junctions that make a nice fence down the length of your intestine. They secrete mucus to the top side, and that mucus can trap bacteria, it can trap toxins, it can trap other things that are in your food that you don't want to take in. What this does is it compartmentalizes everything in your body. So what you ingest stays in the lumen of the intestine, and it's only taken up when your body says, okay, we want that. And then it'll use typically receptors to grab that, pull it in, and deliver it into the bloodstream. Now, this can break down, and it breaks down in every one of us every day to some varying amount. And what happens is these cells start to pull back from each other, and this opens up the ability for things like toxins and for bacterial particles to slide through and get into the bloodstream. And that can cause inflammation at the level of the gut. And as that damage continues, you can get growing areas of inflammation. But those particles and toxins then are also traveling through your body, and they can have impacts on your brain. They can have impacts on just inducing systemic inflammation throughout your entire body. So luckily, members of our microbiome tend to seal those back up, take care of these issues, and can help to regulate a lot of the issues that we see. But I would say that what's happening in the gut with dysbiosis and things like leaky gut and this barrier issue are paramount to a lot of the issues that we see in disease and in unhealthy physiology in general. We know that when you're stressed out, you have anxiety or sleep issues, that gut barrier starts to open up and you start to get some of this recurring inflammation. This is also something that we see very commonly in diseases like diabetes and others like that. So because the gut affects other organs and the microbiome plays such a crucial role in the gut health, it follows that the gut microbiome is directly involved in impacting various parts of the body. And we call this the gut axis connectivity. And I think we hear a lot about the gut-brain axis, but there's been some really interesting papers that have come out recently on the gut-skin axis and where cuts in the skin on the outside can actually have impacts on what's happening within your gut and vice versa. So these are bidirectional signaling. And these are really occurring with nearly every one of the organs within your body. And so it's important to think about that. Because what this does is it opens up a really big opportunity for us to be able to deal with whole health. So the health of the microbiome has far-reaching effects that can impact not only things like mental health, but others as well. So we know that there's a lot of research right now looking at brain health as it's linked to the microbiome, mental clarity, cognitive abilities, but also emotional health, being able to deal with stress and anxiety, and even sleep. And then we're seeing this with metabolic health as well, and there's a lot of work that's going into looking at that, but also digestive health, skin health, and respiratory health. So it used to be that we would look at the microbiome and think it's really only having an impact on the digestive system, and maybe it's having some impact on inflammation, but we're now finding that it's having impacts throughout the entire body. The reason that we're able to do that is that we're able to really advance our science and our studies of the microbiome through more current biotechnology that's going on right now. This is a really exciting time for biotechnology. We have all sorts of new tools that are at our fingertips that allow us to really make pretty extensive advancements, not only in understanding our own physiology, but how it interacts with other things as well. Some of the advancements that we're currently looking at as an industry are combining things like bacterial strains with other things like grains and other botanicals to increase their efficiency. So, I think as an industry, we've kind of been a little clumsy when we first started with this. We would take some, a botanical that we knew had some sort of an impact. We would combine that with a probiotic that had some sort of an impact, and then we would hope that together they would actually increase that benefit. What we're finding now is that we can look at a lot of these botanicals that are out there. We can look at the phytochemicals that are present. We can look at, just the raw materials that are available, and we can begin to really target their use with other biotics. We can take bacteria that may come in and clip them off to really small bioactive components that can then be absorbed into the body and circulated as well. The next is that we can actually begin to utilize precision fermentation, and we can cause bacteria to begin to produce specific metabolites that we know have a target within the body. We can then potentially, if that is a heat-stable metabolite that's being made, we can pasteurize that, and we can use that as what's called a postbiotic. And this is a new way that really takes us away from some of the main issues that we had by using probiotics. So initially, when we had to worry about keeping probiotics alive, being able to put them on the shelf for a couple of years, being able to have them, go into a cup of coffee or something like that. Not something that's very easy for a live organism to do, but if it's specific metabolites that we're looking for, we can now remove that. We don't have to necessarily worry about keeping them alive. We can express those metabolites, and we can use those metabolites then as a new ingredient that can be added. So that's a new area that's advancing right now in the advancement of postbiotics. The other is that we're looking as an industry at developing solutions that deliver functional redundancy. And I think that's important to think about because many of the health issues that we have aren't due to some single factor. It's usually a network of different factors that all come together to create some sort of an issue. And so it might be that I have some mutation in a specific receptor for a vitamin and you don't. And so when we take that vitamin, you get a benefit and I only get really half of that. And so maybe there's something else that would be beneficial if I took it alongside that. And so as we start to pair different ingredients together, usually maybe different bacteria or botanicals along with bacteria, we can really provide a more broad impact across the population. So not only am I getting a benefit, but you are as well. And that allows it to be more functional across the overall population. Finally, we're able to intentionally develop and to deliver metabolites for specific health outcomes. There's been a lot of research done in the last even 5 years to really look at metabolomics and understand what exactly are the active metabolites that are out there. And so we can begin to look at specific bacteria that can produce these specific metabolites. And now we know how we should really be using those when it comes down to clinical trials. And then the last thing that I really would like to talk about is that we can apply biotechnology and advanced science for strain discovery and improved efficacy. And that's really, we're at a very exciting time, I think, in biology right now, because we have not only the age of AI and understanding large groups of information, but we have the age of robotics as well. So we no longer have to stick grad students in a lab somewhere and make them work day and night. We can make the robots do that, which is far easier and in some ways depressing for those of us that had to work in the lab all through the day. But the advances in science have really allowed us to start to deliver more functional ingredients. One is we can combine strains, grains, and botanicals to really make better efficacy. Okay. So I think it's also important to look at an understanding of strains. So it's important to understand that strains do matter. One strain doesn't work for all products or for all health areas. And I think it's important to not just replace one or think of one strain and think that it can do everything. So if you have one Lactobacillus plantarum, you really can't replace it with, say, another Lactobacillus plantarum because they have different functionality. A good way to think about this is to really look at dogs. So dogs are all the same genus and species. They're all Canis familiaris. But if anybody's had a dog, you know that the strain can make a huge difference because there's a big difference between how you're going to use a Chihuahua and how you're going to use a Great Dane. And the same is true for a lot of these bacterial or probiotic species that are out there. Strain makes a huge difference because they can produce different metabolites, they can have different interactions, and they can have actually very different genomes as well. It's important to look at how you're going to use them. There are different diets that are out there, and some can be really difficult to live in. If you're looking at something like pomegranate, it's a low pH environment. There are a lot of antimicrobials that are present, and that's very different than if you're going to put something into, say, oat milk, which is more neutral and is a little bit more friendlier. It's also important to think about where do you need the site of activity to be. Is that going to be in the stomach or is that going to be in the colon? The time of day that you're administering it, because your physiology is changing over the course of the day, and bacteria will produce different things during the course of the day. And then finally, when you're starting to formulate different products, it's important to understand whether they need to stay alive, in which case they may have to be refrigerated to stay alive, or maybe they're a spore former and they can be kept at ambient temperature or they can go into hot liquids. So these strains can all have different functionality there as well. And I think one of the crux and one of the things I've touched on a number of times today is that there are function-specific metabolites and they have a direct impact on the body. The gut microbiome produces beneficial metabolites that account for at least half, and actually, again, newer estimates suggest that 70% of the metabolites that are coursing through your veins right now are a result of the microbiome that you have. And so they have a direct impact on your overall health. What kinds of things are they producing? Well, we know that there are neurotransmitters that are released by bacteria, things like GABA. We know that lactic acid bacteria and bifidobacteria will produce GABA. And in fact, we've been able to go back and find strains that produce clinically relevant doses of these at maybe a 1 billion CFU dose. And so it's important to understand what's being created. Other things like butyrate, which is really one of the keystone metabolites that has a very strong impact on our body, is also produced by bacteria. So butyrate is a short-chain fatty acid that is the main energy source for those epithelial cells that are in your gut. Butyrate also is involved in kind of regulating the cytokine expression, so your immune response. But more recently, it's come to interest because it actually is involved in expression of GLP-1. So it has direct involvement in metabolic history as well. And then there are microbe-specific metabolites like urolithin. The only place you're going to find urolithin in nature is when you have bacteria that are breaking down ellagic acid or ellagic tannins into this metabolite. And I don't know if anyone's familiar with urolithin, but it's a really interesting metabolite. This is a metabolite that when it's released, it goes directly to the mitochondria, so the powerhouse of the cell, and it measures the ability for the cell to be healthy. if the mitochondria is old and kind of senescent, your lecithin will kill it off and induce the cell to make new, fresh mitochondria. So this has implications in muscle mass, muscle growth, senescence, and other things. And it's only produced by certain members of the gut microbiome. And that leads us into kind of the bigger family. I think a lot of us have heard about polyphenols and their metabolites and how interesting they are and how good polyphenols are for your body. If you dig into the literature, you see a large variance to people that can respond to polyphenols. And the reason is because polyphenols are relatively large molecules that have a charge to them. So when you eat them, they tend to stay in your gut. They can get out a little bit, but not a lot. But we found that as you look at some of these clinical trials and the research that's been done, the people that get the most benefit typically have bacteria that break these polyphenols down into simple phenols that are absorbed, they're much more bioavailable, and they can get to the site within your body that you need them to get to. And so these all open up kind of opportunities for ways of developing and targeting new types of probiotics that we can move forward with. So the microbiome, currently, we're at a spot where we can really move our science from labs and into actually changing people's lives. It's a very exciting time. I don't know if everyone's heard, but AI has become a big thing, and metabolic modeling. And this sort of a technology is very ripe for microbiologists and bioinformatics scientists to be able to leverage these genome data, these vast, huge files of genomes that we have to really understand how does a probiotic work. We used to have to take a guess at that. Now we can begin to model that out, which is really exciting. The other is that, like I said earlier, we have robots at our disposal. So we can do things like high throughput screening in a way that we've never been able to do before. We've been able to leverage some of the other resources that are out there in the industry to be able to test 250 different variants, candidates, and isolates to find that needle in a haystack that creates the metabolites that we're looking for at clinically relevant doses. So that's all something that we can do now that we couldn't have done even 5 years ago. And then finally, when you're doing that, sometimes you'll find that an organism does exactly what you want it to do, but it may have some issue, like it creates a toxin or it has an antibiotic resistance gene in it. And now we can use genetic engineering to go in there. And if we need to, we can pull out those genes and get rid of them. Or if it's something that's, maybe beyond repair, we can pull out pathways and put them into a friendly chassis and be able to move forward with those as well. And to now be able to make really safe organisms that we can move forward with. What this allows us to do is then de-risk human clinical trials. So instead of going in and taking a guess at whether or not something's going to work, we can now use AI to build digital twins of people. So everyone in this room could have a profile. And we could take different genomes from probiotics that we want to use and see how they can interact with this other huge network of what's going on within the gut. And that allows us to run multiple iterations to see, is it most likely that we're going to get the endpoint that we are looking for? Are there some new things that are out there that we may have missed, or is this not going to work at all? And so by using this side of a new technology, we're able to de-risk those clinical trials, because that's the number one expense that we have as we develop some of these probiotics into actual ingredients. That also allows us to make much more targeted structure-function claims as well. So overall, the new technologies that are out there have changed the way we develop our products. It used to be that we would start off by looking at strain discovery, and we would get a bunch of candidates, and from there we would immediately move into in vitro bench work, and from there we would take those candidates and we'd go straight into clinical trial validation, and we would hope that these would work out. But now, because we have some of these new tools, we can go from strain discovery, and we can now vet them using modeling and different systems to understand, do they produce the metabolites that we need, and how much is being produced? And from there, we can then move into in vitro bench work, confirm that they do what we expect them to do, at least in culture. We can expand out on their mechanism of action and really dig in to understand what they're doing and how they're doing it. And then we can move that into looking at how do they function in a complex environment. So we can now take different databases and we can look, when we add our probiotic into the system, is it the lion that we expect it to be? Does it create the metabolites that we want? Does it grow the way that we want? Or is it a lamb? Does it hide in the corner and everybody's just stealing its lunch, beating it down, and it can't grow and it can't succeed? And we can start to do that sort of modeling right now, which allows us then to get better candidates that we can move forward into and move into these clinically, the clinical validation stage. So I'd like to stop there. I definitely want to hear what Mike has to say about commercializing some of this, but I hope at this point you have an understanding of what the microbiome is, how it affects other organs within the body, and then also some understanding of how biotechnology is really moving things forward, because we're at a space now where we can truly make functional metabolites and functional ingredients that can be used for whole health and whole body health overall. So thank you.
That's right. Yeah, thanks. That was great. I want to invite everyone. You can continue to ask questions. We got 4 that we've received in already. And one of them, I think, already got answered. It was asked earlier, and it was about AI, the use of that. And I think Noah explained that pretty well. So now I'm going to introduce you to Mike Bush. Now, Mike Bush, let's see, you cut your teeth a long time ago. I'm just going to go with Ganeden BC30, which was kind of the OG spore-forming probiotic, which had the great benefit that you could use it in foods and beverages, and it could just withstand processing. So it just improved the survivability of probiotic strains. And that company has been acquired. And so, but Mike, as the president of Ganeden, and he worked there for a number of years, he helped usher the company through that whole process. Now Mike has hung his own shingle, GrowthWays Partners, where he's a consultant with other brands and getting them to where they need to be. So after Mike finishes, then we're going to have Q&A, and we're going to talk about some of the Q&As that came in for Noah here. So Mike, take it away.
Hello. Before we get started, as Todd said, I've been in the microbiome space since 2006 or so. I joined Ganeden when they were a consumer products business, and I joined them to start an ingredient business that we call Ganeden Labs. Before that, I was in healthcare, which was incredibly depressing, and so I wanted to get into the let's keep people healthy space rather than let's make them sick and charge them until they're bankrupt. Previously, I was the president of the International Probiotics Association board, and I was on their board for a very long time. We measure board terms at the IPA in horse years. And as Todd said, I was the president and CEO of Ganeden. We sold that to Kerry in 2017, and Kerry continues to bring the BC30 brand to market. It's been an incredibly successful story. It was a great sale, and Kerry has had really, really good luck at keeping things moving. There's thousands and thousands of products around the globe that contain BC30. And so when I left Kerry and decided that I was just going to kind of consult and help build value, I formed GrowthWays Partners, mainly as a mechanism to share the kind of experiences I've had. Ganeden was the 7th company I was part of the sale of. I don't know anything but how to grow a business to be sold, or to grow a business to be made more valuable. And so as a show of hands, who in here is in the microbiome space? All right. And who in here is in the space where your kind of mission is to grow a business to build value that ultimately may be sold? It's a family business, you want to grow value in it, or you're going to go public. I mean, one of those 3 options. And the rest of you must work for not-for-profits, because that's really what you do with a company. You grow it to go public, you grow it to sell, or you grow it to, to continue to generate value for generations. And so that said, just to wrap up, I've got a book coming out in June that talks about how to build value in any business. It's called Guardrailing. The title that we finally settled on yesterday is Guardrailing: How to Authentically Guide Your Natural Products Company from Spark to Sale. Feel free to see me afterwards, and I'll get you on the mailing list so we can notify you when the book comes out. There'll also be a course that goes with it and stuff. But anyway, value is incredibly important. So when we look at value drivers, people sometimes think it's, well, we have all this IP, we're valuable. Or we have an influencer, we're valuable. But there's a lot of things that involve value. And so kind of to take it in no specific order, you really need demand for the product. You need your product to be unique in the market. You need to have scarcity of your product. If you sell something that everybody else scares, how much value is there really? You need to have benefits of that product to the buyer. As part of my whole guardrailing mission, it is, are you doing good? Guardrailing consists of two questions. Does this decision build value long-term in our enterprise? And the second question is it a good, option? If it's not doing good, if you're not doing good, you're not building value. And when I say do good, it means good to every stakeholder, to your suppliers, to your vendors, to your partners, to your customers, to the end consumers, to the family members of your end consumers, to your employees and their families. You have a responsibility to make good decisions, ethical decisions. Obviously, as Noah has been talking about, you need data to support your decisions and the benefits. You need to protect against competitors, obviously. And one of the first things you can do to build value, especially in an earlier stage business, is to find somebody like Noah who can bring accessible science to the masses. The stuff that Noah was talking about is incredibly complicated. He made it seem simple because that's what he does. But you really need somebody like that in your corner so that you can distill the science down and show the value. So when we talk about building value in biotics, as I said, I was with Ganadin, and we had all kinds of different iterations of our model. Harvard actually wrote a case study when we switched from being a consumer products business to being an ingredients and licensing business. And it was really interesting how, when we go back and read that case study, that was right when we were starting the business, we pretty much nailed it. If you look at the path, it was like a really nice linear path of growth that was secretly, we made every mistake there was known to man. We had regulators hating us. We had retailers hating us. We had all kinds of things happening, but we felt we were making good decisions for the right reason. That was to support consumers, and so we didn't care. But when you're building value in biotics, you have to own it, right? So many companies, they're out there putting out Me Too products. I mean, that's great if your goal is to generate lots of revenue and sell the business for some multiple of revenue. But valuations in this particular industry go, let's say, I'm not an investment banker, but multiples have been right around on average around 13 to 15 times EBITDA. And so if you want to sell your company for $100 million, it's easy math. You know what your EBITDA needs to be in order to be in the average of your multiples, and you grow your business to there. How hard is it, right? So one of the things that I hear from every biotics company is about their new patent. They got a new patent. I saw one the other day. It was a really great patent. I mean, really great patent. It had really good claims. It supported all kinds of really neat things, and it was on that strain. So no one could use their proprietary strain that no one can use anyway. And so they built all this IP around this strain that only they have. I mean, I realized in the future people can go out and grow their strain and things, but their patent is so specific to their individual organism or their individual ingredient that it's kind of like you feel good. It's kind of like the people on Shark Tank. They walk in, they're like, I got a patent. They go, oh, that's awesome. You got a patent on something no one will ever use. So you want your patents to be as broad as possible, but trademarks are the first line of defense in the biotic space. In particular, those of you who are in the probiotic space and you may own strains. You've discovered a strain, you've developed it, you've put it in the ATCC, you've done clinical studies, but you didn't protect your strain name. So the folks out there that have a similar product are out using your clinical data saying that Lactobacillus plantarum, whatever, does this, and they're using your data. It happens all the time in this industry. I mean, I would be shocked if anyone in this industry doesn't know of someone or isn't doing it themselves or hasn't had it done to them where they're borrowing science from similar ingredients. With a trademark, you trademark your entire strain name. So in the case of the Ganadin BC30, it was Bacillus coagulans GBI 306086. We trademarked that name. We trademarked it everywhere. Now, no one can use the clinical studies as a reference on their website. So say Todd starts, whatever, biotics, and they decided that they wanted to show that their strain of Bacillus coagulans did this and that, and so they hung out, the references to the GBI 30 strain. Well, they would be in effect violating our trademark, and they would get a quick call from the attorney. So you really need to make sure that your science is not borrowable. Because as we know in this industry, we've known this for a very long time, and those who don't admit this or fool themselves, everything's strain specific. So if you're selling Lactobacillus acidophilus 1, 2, 3, 4, and you have data that it does X, Lactobacillus acidophilus 3, 4, 5, 6 may not do that at all. It may do nothing. It may do the exact same thing. But until you have the clinical data on the individual strain, you don't know. So your value is being built through all these moats. The technology name. I'm a big fan of naming services and technologies. I just think that that people can relate to brands rather than very, very complicated things. You obviously need to establish your product's unique benefits and promote them. So you want to have published studies. I had an investor one time that at a board meeting, he literally slammed his fist down on the desk and said, "When are we going to be done with all this clinical stuff? The science BS has to stop." And I said, never. We have to continue to do science and look at clinicals and look at the latest science and do new clinicals. And so by the time we were done, we had 37, 38 published clinical studies on our strain because you had to do that. You had to keep up. And then patents obviously is a necessary moat. In the world of Verb and Ginkgo, ultimately there may be genetic modification happening. It's going to be happening in our space. Zbiotics has a GM organism that's out right now. It's I think it's the first one to hit the market commercially, but it's a genetically modified Bacillus subtilis that helps in alcohol metabolism. So those patents on those strains that are super unique, you can still patent strains, but now you can't patent organisms just for the good old days when you could just patent living things and say I own that. And so patents are nice, but they're defensive moats are primarily around utility. So if you have an organism that can go into a particular application and you're the first one there, patent that application and go as wide as possible. Ultimately you want to be genus and species if you can when you're doing patents rather than your strain. If you could get genus, you've got the most brilliant patent attorney in the world, and I would like their name. But genus and species is pretty powerful. So really the 3 P's in value are you got to protect it. You got to own everything. I'm not a big renter when it comes to things that I ultimately want to sell. As you can tell, that kind of doesn't go well together because if you're renting an apartment and it's the coolest apartment in the world, your only option is to sublet it. You can't sell it. So if you're renting your technology or you're licensing with some sort of expiration on a license or some sort of term that makes it not perpetual, you can have that technology go elsewhere if you don't own it. Obviously, file your patents, your trademarks, and maintain publication rights. Please, when you do clinical studies, maintain your publication rights to add value. I just recently, and I won't name names, but a $1 million plus clinical study was run recently. And it was done by a university. The company did not maintain complete control over their publication rights, and they had this paper published that had, say, like 4 endpoints that they're looking at. 2 of them were knocked out of the park, and 2 of them were meh. And so, but human nature is human nature. And so, what did everybody do? They looked at the clinical studies, and they said, it's a meh. It's a meh. It's an average meh. And so they didn't get the bang for the buck because they didn't really maintain the publication rights. If they would have done it right, they would have run a pilot study that would have said, these 2 endpoints may not work, so let's not spend $1 million and include those 2, because in the publication you got to talk about the whole study. You can't just talk about what worked. Promote it. Obviously, please follow the IPA's labeling guidelines, strain, genus, species, strain designation, CFU and milligram or CFU or milligram. When it comes to postbiotics, you're a milligram, same thing with prebiotics, but make sure that you're using the right, verbiage. Publish all your data, publish everything. Proprietary data really does you no good when it comes time to build value in a business. And make sure with finished products, if you're an ingredient company, the greatest get is to have your logo on the front of pack. And so that's the get. If you can have your logo out on packages, And I remember in the early days of Canadian, our very first customer that we asked them to put the logo on the package, and they were signing this document saying they would do it, and they would follow all these rules. They said, "How much are you going to pay us to, to put your logo on our package?" And I said, "Well, that's the funny part, because it's actually part of the price that you're paying us. So we're giving you the right to do it, but we're not going to charge you any extra." And he's like, "But it's already included in the price, right?" And we're like, "Uh-huh." He's like, "Well, what if we don't want to use it?" And I go, "Well, then we can't sell you the ingredient, because that's just the way we work." And so we had the logo on the front of the pack on our very first product we launched, and it was on about 80% of the SKUs that were launched around the world by the time we sold the company. We were in 1,400 SKUs, sold in 70 countries, and it was about 80% of the SKUs that we were in had the logo somewhere on the package. It was incredibly helpful because it's one of those things that people just start recognizing the logo. At one point, we had 58 SKUs in the average Whole Foods store. So you'd walk through there, and people were really shopping, and they were looking for the BC30 logo on their tea and on their whatever. So do that if you can. Promote the brand, obviously. Establish systems to monitor your trademarks and your patent infringements. Fortunately, it's a small world, right? So Google Alerts is the simplest way to do it. You could hire, trademark and patent infringement monitoring companies, but really Google Alerts works pretty well. And actively enforce them. And so only in one occasion did we have to even send a letter. It was usually, "Hey, Charlie, I just looked at your website and you're using our clinical studies again. Could you take it down? I really don't appreciate it. Thank you. Buy you a beer at the next show." It was kind of those type of conversations. Only once did we have somebody refuse to take something down, and it came down a couple days later. But you really have to actively enforce it, because if you don't enforce your trademarks and you don't enforce your patents and you knowingly let people do whatever they want with them, then they can be invalidated. So fight for yourself. So just real quick, we're going to do a little assessment here. So if these products, and these are two products that are on the market, which one could generate the most value for the company? Is it the one with a probiotic blend with Acidophilus, Bulgaricus, Bifidum, and Thermophilus? Who knows how much of each one of those is really in there, because that was at time of manufacture. You can't see that, but it's at time of manufacture claim. But which Acidophilus is it? Is it a good one, or one that they found they dug up out of some generic acidophilus.com website? Same thing with the rest of the strains. While on the other one, which you may recognize as Culturelle, Lactobacillus GG, 10 billion CFU, and that's at time of consumption. They really put, I believe at the time, they were putting 30 billion CFU in at time of manufacture to get to 10 billion at time of consumption. And then they had some inulin in there, 200 milligrams inulin. Honestly, I don't know what 200 milligrams of inulin does, other than it does say inulin, and people like having inulin with their, as we saw earlier, people believe that you need to feed the probiotics with the prebiotics. And so, but when you look at this, the way they labeled this, there's just added value in the fact that this has Lactobacillus rhamnosus GG in it. A consumer can go out and look up the clinical studies related to GG. For the first one, you have no idea what's in there. Can you look it up? You can find out if those genus and species are okay, but that's as far as you can get. So you can make no good decisions based on that label. And so I don't know why any brand owner would label their products this way unless the reason is to intentionally confuse consumers, or to leave the door open so that when they can save 2 cents a kilo on a Lactobacillus acidophilus, they can do the old switcheroo without having to reprint labels. And and the question I have is that a good decision? Is that a right decision? No, it's not, because it's not giving consumers what they believe they're getting, which is a clinically studied probiotic. And this goes across the board, whether you're talking prebiotics, probiotics, botanicals, whatever the case may be. Put in there what you say you're putting in there, have the data available, and make sure that you're protecting it so that as you go forward, when your company's in due diligence to be sold, or when you're going to go public, or when you're doing a board meeting and you want to see what's happening to the value of this business, you can actually point to these things and say, we know that these particular decisions that we've made over time have created additional value in the business, so therefore we're doing things the right way. And in the meantime, we've made the world a better place, we've made people healthier, and we haven't done anything wrong. So with that, I will sit down and we'll take questions.
Thanks, Mike. That was bloody brilliant. I know I feel a little smarter. We did receive a number of questions in here, and I want to start with the science. Noah, with you, a couple of these came in that are interesting. One is what current evidence supports the notion that many exogenous probiotics, that would be the supplements, they do not permanently colonize the gut microbiome, and what's the implications for their therapeutic value?
Sure. So I think there's the microbiome is pretty well established within a body. By the time you're an adult, it's pretty stable. Now, there are some fluctuations that occur, but it's very difficult to introduce a new strain that's going to colonize, take up residence, and be able to compete with everything else that's worked out in that system, mainly because it is a system, it's a network. So typically what that means is you will need to redose over time. So we've done clinical trials where we've monitored, can our strain, do we detect our strain after so many weeks, and do we detect it, over time? And there are also animal studies, because probiotics are being used in agriculture as well. And we do see that typically they do not colonize within the gut because that system is already within balance. Even when you have things that are out of balance, when you add in, they tend not to work or colonize across time. And in my opinion, that's a good thing, because you don't need the same bacteria throughout the entire course of your life. This needs to change over time because you're eating different things, your body is responding differently, your physiology is changing. And so your microbiome needs to change with that as well.
Yeah, so a question came in, and it relates to something that you said, Mike, which my bells rang off on my head, which you said, I don't know what 200 milligrams of inulin does. So, but that's kind of the conventional wisdom out there, right? That you need the prebiotic lunchbox to feed the probiotic. Are you saying maybe not?
Maybe not. Or my big question, whenever I see somebody matching up a prebiotic with a probiotic, I say, thank you, person, for matching up this prebiotic with this probiotic. Is this the ideal carbon source for this particular probiotic? And how do you make sure they work together? And how the heck do you make sure that 200 milligrams of inulin finds its way into these, to feed just these, 1 billion CFU of Lactobacillus rhamnosus GG? How do you know that? And usually it's like, consumers are looking for prebiotics too. It's kind of like one of those, I've not gotten a good answer for that. And I haven't seen any really good clinical data on these combination products. I have seen good data on custom carbon sources being used by companies like, you've got companies out there like, I can't remember some of the names right now, but they're making custom prebiotics that go specifically with a probiotic. And so if you're doing that, awesome. If not, stop trying to fool people.
Right. Well, because it seems like if you want a prebiotic for the fiber, say, then we're talking grams. We're talking 4, 6, 10, 15 grams, right? But as a feed source for the probiotic, then maybe you can get away with milligrams. And I remember like 10 years ago, we were saying, okay, one day we're going to be able to match up the pre and the pros. And so you're saying we're not quite really there yet?
I think we're there yet. I think we're there. There are people that are making, custom prebiotics that match up, and so you know that you need this much to go with this organism, but they're not doing it. I mean, Noah.
Yeah, I would agree, and I think that part of it is that it's expensive to get good specific prebiotics, but the other is that it's an entire system again. So it might be that you don't need to feed the probiotic you're putting in; you need to feed something that's going to feed the probiotic you're putting in. And so it's a much more complex system than I think initially thought. It's not one-to-one. And that's where the new technology is really making a difference, because you can begin to look at how does AI look at this. You can try millions of different iterations to see how does that work out.
Yeah, and so speaking of which, one thing, Noah, that you had said was you're using high throughput screening, and you've been able to get through, you had said 250 different strains. You had meant to say 250 million different strains. I mean, that's the mind-blowing thing that we could not even imagine 5 years ago, and now that we're able to use AI and high throughput screening to really, and get through stuff at a speed that's unheard of, So I think that's really interesting. Another question came in similar to that one, which is what is the use of a postbiotic? What is it exactly, and how does it support probiotics? Is there a relationship there?
Well, there is and there is not. It depends what it is. So I think right now, sometimes we get stuck in what the definition is. A postbiotic means that you have something that has a health benefit, but it's not alive. That really means that for that to be a functional postbiotic, you better know how it works. You can't just take a probiotic, kill it, and expect it to have a function. So when we've developed these, we look at what metabolites are being made by a postbiotic or by some microorganism. Are those metabolites stable? If they are stable, then the organism doesn't need to be alive. We can take those metabolites and part of those organisms and use that as a product. Sometimes they will directly feed other probiotics. Maybe, a postbiotic can have prebiotic effects. But many times, if you have the right postbiotic and you have the right metabolites there, they will have a direct impact on either other organisms or on the body itself.
Cool. Can you share an example of a pro or a prebiotic that's being produced through precision fermentation, and what are the economics of that?
Well, I sure can. So Verb Biotics has one if you'd like to stop by our booth. The idea is we've taken something like a specific, we used oat flour in particular with 2 specific strains of lactobacillus. And the idea was that we knew that the oat, because of the phytochemicals that were in it, induced expression of specific metabolites from the probiotics we were looking at. And so those have then gone into clinical trials, and they're performing as would be expected because we know what metabolites are there. We know what the end goal is of those metabolites. And so by using that precision fermentation, we're able to really direct the use of, or target, I guess, the function of that ingredient.
That's pretty crazy. This might be the last one, but when combining grains and strains, what criteria should be used to select the right type and quality of the grains?
What is the phytochemical that you're looking for? Because if you grow a field in Oklahoma, you may have different phytochemicals growing in your wheat than you would if you had it growing in Iowa or Wisconsin. And so you really need to, again, look at what is the overall phytochemical profile, or what are the nutrients that you expect to have some sort of an impact, and then really focus in on those.
And this just in, I think I know the answer, but can we claim prebiotics in a formula that contains only 200 or 300 milligrams of an inulin or a pectin or a starch when we know we need grams to claim it?
I say no, but that's the F-sub, compliant person in me. But my
if you want to be a responsible brand,
I want to be a responsible. Yeah, I'd say if you want to be a responsible brand, if a prebiotic has a definition, it has a benefit to a host, just like probiotics. We all know that probiotic in Europe is a claim, right? So prebiotics, by using that same rationale, if all of the clinical studies on a particular prebiotic are at least 5 grams per day, then it is not effective unless you are taking 5 grams per day. I saw just recently a supplement that had 32 milligrams of inulin in it, and I couldn't help myself. From the floor of Target, I called the CEO of the company, who I know, and I said, "What in the hell are you doing putting 32 milligrams of inulin in the product and calling it prebiotic?" And he said, "Marketing." And I said, "Marketing people should be fired." Simple as that.
There you go. 45 seconds, lightning round. Last question. Explain why it's not as important to keep the biotic alive.
It's important to keep it alive if it needs to be alive. If it's a postbiotic and you know the metabolites, as long as they're there, you can get a function.
Yeah, but probiotics you do need to keep alive. But and a probiotic doesn't become a postbiotic when you've consumed it. That's just
correct.
A not live.
So typically you need to have the probiotic alive so that it's a factory, right? It's making different things. That factory needs to be active, or else there's just nothing there to work.
All right. Thank you, everybody. Noah Zimmerman, Verb Biotics, Mike Bush, GrowthWays Partners. I'm Todd Runstead, naturalproductsinsider.com.
One last thing from the back of our room. If you haven't taken the survey that was on your little VBox Q&A next to it to the right, please do. We appreciate all your feedback. Thank you so much.
There you go. Cheers, people.